Hello. So we're continuing with life science and biology in a nutshell. Um, Currently we were learning about the endoplasmic reticulum. We learned about the rough endoplasmic reticulum. These are organelles or parts of the cell. So smooth endoplasmic reticulum is different from the rough endoplasmic reticulum in that it doesn't have ribosomes, which are used to ribosomes are used to as the site for protein production off of mRNA templates, which come out of the nucleus after DNA is transcribed. So genes are transformed into proteins or polypeptide chains in the ribosomal subunits of which lie on endoplasmic reticulum folds. However, in the smooth ER, there is no ribosomes. Smooth ER, in contrast, is associated with the production and the metabolism of fats and steroid hormones. It is smooth and it is not studded with ribosomes. It is only associated with smooth, slippery fats. It is more tubular and it forms an interconnecting network, the compartment of the ER. It is found fairly evenly distributed throughout the cytoplasm. Smooth ER is devoted almost exclusively to the manufacture of lipids and, in some cases, to the metabolism of them and associated protein products. In liver cells, for example, smooth ER enables glycogen that is stored as granules on the external surface of smooth ER to be broken down to glucose. Smooth ER is also involved in the production of steroid hormones in the adrenal cortex and endocrine glands. And so here's the folds of the ER, as well as ribosomes, which are the darker dots, but the smooth ER doesn't contain ribosome. It's just cisternae, which are the folds. The endoplasmic reticulum, that's what it kind of looks like. It starts off from the membrane of the nucleus and it goes outward towards the cytoplasm. Smooth ER also plays a large part in detoxifying a number of organic chemicals, converting them to safer water-soluble products. Large amounts of smooth ER are found in liver cells, where one of its main functions is derived from excess alcoholic drinking and also for detoxifying products of natural metabolism and endeavor to detoxify overloads of ethanol from excessive consumption of alcohol or barbiturates or drug overdose. To assist with this, smooth ER doubles its surface area within a few days, returning to its normal size when the assault has subsided. The contraction of muscle cells is triggered by the orderly release of calcium ions. These ions are released from the smooth endoplasmic reticulum. Besides lipids and oils, SCR also create and store steroids, which are a type of ringed organic molecule used for many purposes in an organism. Both types of endoplasmic reticulum are present in plant and animal cells. The two types often appear as if separate but are subcompartments of the same organelle. Cells specializing in the production of proteins will tend to have a larger amount of rough ER, while cells producing lipids and fats and steroid hormones will have a greater amount of smooth ER. And I'll put in what the steroid molecule looks like. Let's see. ER works closely with the Golgi apparatus, ribosomes, mRNA, and tRNA. The ER and the Golgi complex are so close that some chemical products probably pass directly between them instead of being packaged into vesicles or droplets enclosed within the membrane and transported to them through the cytoplasm or through them. The ER membranes are slightly different from cell to cell, and the cell's function determines the size and structure of the ER. For example, some cells, such as prokaryotes or red blood cells, do not have an ER of any kind. Cells that synthesize and release a lot of proteins would need a large amount of ER. However, you might look at a cell from the pancreas or liver for good examples of cells with large ER structures. The Sarcoplasmic Reticulum, or SR, is a variation of the smooth ER. It is able to store many ions in solution that the cell will need at a later time. When a cell needs to do something immediately, it doesn't make sense to search the environment for extra ions that may or may not be floating around. It is easier to have them stored in a pack for easy use. For example, when you're running around, your muscle cells are active. They need calcium ions. The Sarcoplasmic Reticulum can release those ions immediately. When you're resting, they're able to store them for later use. We have a diagram here showing the dimensional 3D endoplasmic reticulum, the rough and the smooth, that start out from the nucleus outward. Staining of endoplasmic reticulum in human cell lines. Immunofluorescence staining shown here. As the ER is contiguous with the nuclear membrane, it is recognizable by staining of nuclear membrane and a network-like staining of the cytosol. It is usually stronger close to the nucleus and weakest as it gets closer to the edges of the cell. The network consists of thin strands that intersect at regular intervals. So this is a beautiful immunofluorescent photo. And here again, the nucleus in blue and green. You have endoplasmic reticulum outside the nucleus. Colored transmission, electron micrograph, or TEM, of rough endoplasmic reticulum, showing a long membrane-bound vesicles in gray, bearing ribosomes in the dark brown spots on the outer surface. The membrane of RER is contiguous with the outer membrane of the nucleus, or the nuclear membrane. So again, you can see it here as well. The function of RER is to assemble amino acids into peptide chains, which with further processing form proteins. Rough ER is present inside most cells, and its abundance is proportional to the level of protein synthesis without the cell. Magnification 25,800.